Thank you. Gosh, I realize I need to have a new mugshot for the web, right? But be that as it may. I'm a first timer here too at OIS, and I'd like to thank the organizers for the opportunity today. Let's see if I can. So at Anida, we are developing AP001, also known as neuroprotectin D1, an endogenous protective sentinel that's upregulated in stress situations. We've then shown in various experimental situations that administered exogenously, it has rather profound therapeutic efficacy, perhaps particularly relevant in tissues of neuroectodermal origin. So how does this work? So AP001 is an agonist. With receptor activation, you get multiple downstream pathways activated that will then independently control inflammation, cell survival, but also stimulate tissue repair. And I think that triad may be particularly important in treatment of chronic conditions, even acute conditions, where we have cells which are terminally end-differentiated. A lost neuron is a lost neuron that goes for cone or rod or hair cell too, as we also have a hearing loss program. The molecule is highly potent, and we have made, as indicated by the round spot in the carboxyl group, some minor modification to it. So how does this then work? So with the pleiotropy and downstream activation, we regulate apoptosis, necroptosis, so both to two death pathways here, multiple inflammation pathways, including regulation of oxidative stress-inducing enzymes, as I mentioned, also stimulating tissue repair here. And we do have a pretty good understanding of what's going on behind the scenes here, having mapped pathways in quite some detail, both in vivo and in vitro. And I'm just going to give a few examples of what I think is the beauty of agonism to control pathophysiology, or as we say, physiological control of pathology here. And whenever, for example, in retinal disease and like dry AMD, I think it's increasingly considered the need to target both apoptosis and necroptosis. And as you can see here, we do regulate apoptosis by upregulating a phosphatase that in turn will control the BCL2 family of proteins. But simultaneously with that, also regulation of the anti-apoptotic proteins, as shown here by BRC3, which will then inhibit the extrinsic pathway, but downstream of that, also ubiquitination of WIP1 and WIP3 with control of attenuation of necroptosis. And we see the benefit in a range of models, in vivo, in vitro. I'm just showing 3 examples here. And the one to the right, particularly the most impressive, with complete optic nerve transection, you still have a 3x increase in retinal ganglion cell survival. So then what about regulation of inflammation? And regulation of inflammation, it's not just inhibition, but also active dynamic regulation. And here's an example of that, and that is AP001 turning bad actors into good actors by changing the phenotype of retinal microglia in the laser-induced neovascularization model from pro-inflammatory amyloid to pro-resolution ramified. So you don't want to change the numbers, you want to change how they behave, and as you then see down to the right, you can have a rather profound therapeutic efficacy, and in this case, delivered topically. Further on neovascularization here, and more to stress the importance of agonism. You see to the left, dosing just every other day is quite sufficient to have a profound efficacy in the laser-induced model. In the oxygen-induced, I've shown an example here, treating early for just 4 days. And the purpose of showing this is that when you try to control the events upstream, you retain VEGF. So in contrast to an anti-VEGF compound, you actually have a reduction of vaso-obliteration, while at the same time you control neovascularization, which is in stark contrast to what you see with Eylea as an example here. So then how to deliver this molecule? We know we can deliver it based on its physiochemical properties as an through intravitreal injection, but we have decided to go for delivery as an eye drop, because the molecule has rather unique amphiphilic properties which allows distribution to the retina. So you see in a fairly low concentration, just dissolved in saline, no need for a particular formulation, do we get scleral distribution. We've looked at 7 different tissues here. And we think that is a key factor for topical delivery, that you can have an agonist which just transiently needed to switch on and off various activities. So where are we then taking the molecule? We are with its properties overall, looking at a first treatment in retinopathy of prematurity, with the intent of delivering it as a prophylactic to prevent the progression of the disease. In order to get there, however, we will need to first study an adult population, which will also give us an opportunity to look at DMA and wet AMD. I don't have time to go into this today, but in 4 different in vivo models do we have activity that quite profoundly indicates that we could also go for anterior segment indications, and we can do that with full product differentiation. So I leave you with this final overview slide here. Eye drop delivery of a molecule with rather unique amphiphilic properties to reach the retina, and then the PD-PK dissociation. And we believe with this we can just, and its tolerability, titrate up till we reach clinical efficacy. And as I said, we're now in IND enablement, and next year we will know. Thank you very much.